Hello and welcome to the Ask the Doc podcast. I am your host for today, Dr. Christian Salazar, epidemiologist at UCI Mind. And if you're joining us for the first time, this is a monthly show where we interview leading experts in Alzheimer's disease and related dementia research. Today, we're joined by Dr. Leslie Thompson, who is a renowned researcher and professor in the Department of Psychiatry and Human Behavior in the School of Medicine and Neurobiology and Behavior in the School of Biological Sciences here at UCI. Now, Dr. Thompson received her PhD in Biological Sciences at UCI, and her groundbreaking research is focused on understanding the biological mechanisms involved in Huntington's disease, as well as ALS and X-linked dystonia Parkinsonism, to identify new treatment modalities and strategies. We are privileged to have Dr. Thompson with us today. Welcome, Dr. Thompson.
Hi, thank you, Dr. Salazar. It's nice to be here.
Yeah, thank you. And welcome to our viewers. If you are attending live on Facebook or on YouTube, you can participate in the question and answer box with Dr. Thompson by typing in your questions in that comment box, and I'll try my best to get to your questions. And so Dr. Thompson can answer all of your questions. So Dr. Thompson, what is Huntington's disease, and what inspired you to study this specific disease?
Yes, thank you. So Huntington's it's an absolutely devastating neurodegenerative disease. It is, if you think about maybe a combination of Alzheimer's, ALS, Parkinson's disease, you have Huntington's. It tends to strike in midlife, sort of in the prime of life, when you have you're at your maximum earning potential, you have young children typically, and that's usually when this strikes people. It lasts for 10 to 20 years of progression. It's inherited, so it's a dominant disease, which just means that if you inherit the mutation, you will get the disease. And it's completely penetrant, which means that if you do inherit this, you will manifest disease if you live long enough. And it's characterized by a movement aspect, so choreiform, sort of dance-like movements, involuntary movements, psychiatric symptoms, so major depression, psychosis, changes in personality. That's typically one of the first things that families notice, and cognitive decline, where it's a very specific component where you just can't carry out daily tasks. You don't recognize patterns anymore. It's very hard to take care of your family. And in the meantime, your children are now 50% at risk because it's a dominant disease. So, and in terms of how I got interested in it, I was a graduate student here, as you mentioned, and I took a course in human genetics from John Wasmuth, who was a faculty member here in biological chemistry. And it was at this amazing time in human genetics where the Duchenne's muscular dystrophy gene, the causative gene that caused Duchenne's muscular dystrophy, which is a childhood muscular dystrophy, had just been isolated. Francis Collins, who was the former head of the NIH, had just shown where in the DNA the gene for cystic fibrosis was. And a marker for Huntington's disease had just been identified as well. So we knew what chromosome the gene was on. And I thought this was just incredibly fascinating area of research. And so when I became a postdoctoral fellow, I joined the lab of John Wasmuth and became part of this international consortium to identify the disease gene for Huntington's disease. And once I met families, once I met Nancy Wexler, who's actually in this picture back here. Once I met her, who runs the Hereditary Disease Foundation, that was it. I haven't stopped. And that was, we as a consortium identified the disease gene 30 years ago last month, or in March. So it's been 30 years.
Wow, it seems like you've done so much, and it's very inspiring to hear that there are researchers that you met very early on that kind of inspired you to get involved. Are there any particular populations who might be at risk, at highest risk for Huntington's?
No, this is an equal opportunity disease. It strikes both sexes, males and females. It strikes worldwide. There's some pockets of populations where there's more people who are affected than in other regions, but that's more just sort of how it has evolved. So for instance, in Venezuela, near the Lake Maracaibo region, there's a very large population of individuals at risk or with Huntington's disease. They tend to have very large families. They have them very young. They might have 13 children. So it just keeps passing along the generations and, across the population. So there's pockets that have many, but there's not any kind of real selection. There's some populations that maybe have a little bit lower incidence in areas of, say, Asia and Japan, in Africa, but in general, it strikes across the board.
And those families that you just mentioned in Venezuela and other countries, would you say that that's mainly due to a genetic component, the genetic component?
Yes, it's due to the genetic component passing along the gene in those populations. Like I said, they have large families, and this is dominant, so you have a 50-50 chance of inheriting it. Although sometimes anecdotally we see more than 50% in a given family of the children affected or inheriting the gene. And actually in that picture behind me, Nancy is holding a child with HD. Sometimes it affects children. It's more rare, but it can affect children as well. And she's holding one of the children in Venezuela who has Huntington's disease. And
you mentioned that the disease is part of what we know as, a neurodegenerative disease, right? How does it compare with other diseases like Alzheimer's and that we're familiar with?
So I think one of the key pieces is that we do know the genetic mutation. It's caused by what's called a trinucleotide repeat. So it's a repeating unit of DNA that repeats over and over and over again. And when that expands beyond a certain threshold of the number of repeats, so if you have 40 repeats in a row and above, you get the disease. Whereas with something like Alzheimer's, Parkinson's, most cases are sporadic, though there are some that are what are called familial, and we know the mutation that causes it, but most are sporadic, and we don't know what is actually causing the disease. So that has a couple of implications. One is that you can undergo genetic testing to know if you're going to get Huntington's disease, because we do know the mutation, which has a lot of sort of dynamics that goes along with that, because we have absolutely no treatment that changes the course of the disease. The other point is that they affect different regions of the brain. So in Alzheimer's disease, it's primarily in the hippocampal region of the brain. In Parkinson's, it's dopaminergic neurons in the basal ganglia. In Huntington's disease, it's primarily in the basal ganglia, but it really affects quite a bit of the brain. It affects the cortex. You have sort of shrinking of regions of the brain, including the cortex. So they affect different regions. At the same time, there's a lot of commonalities across the different neurodegenerative diseases. So they all have issues with handling protein clearance, clearing out the trash in the brain, in cells of the brain. They all have changes in RNA and protein production. So what genes are expressed in various cell types. They have changes in the communication between different cells in the brain. So there's a lot of commonalities and then a lot of unique features of these diseases.
Mm-hmm. And we have an audience question here. The audience member wants to know if there are any risk factors.
That's a great question, actually. And it's funny, I was just teaching a class and we were just talking about this, is so for Huntington's, it's not so much a risk factor because we know the mutation, but there are what are called modifier genes. So there's genes that influence when an individual gets the disease. So the time at which somebody starts to manifest symptoms or show symptoms of the disease, which, as I mentioned, is this and typically it's when the movements start, but usually some of the cognitive and psychiatric aspects have happened before that. Most of that, or 50% of it about, is accounted for by the number of repeats. So the longer the repeats, number of repeats in a row, the younger the age of onset. But we know that there are a number of genes through what are called GWAS studies, or genome-wide association studies, where you look at the DNA sequence of individuals who have later or earlier age of onset, and have found that there are genes in what are called the DNA damage repair pathway. So these are genes that can, as we go through life, there's constantly mutations in our DNA or imperfections in our DNA that happen over time. And there are mechanisms in the cell to correct that. And genes that are involved in that process are found to modify the age of onset. So either they don't work as well, or they work too well and have implications to HD. And it's interesting because, say, for Alzheimer's disease, many of the risk genes, or things that would make you more likely to develop Alzheimer's disease, are things that are influenced by what are called microglia or immune responses, or ApoE4, things like that influence if somebody might get Alzheimer's. So we know with HD that someone will get Huntington's. It's just a question of when, and those genes there's genes that influence when.
Great. And talking a little bit more about mechanisms,
Yeah.
as you point out, we've learned a lot. I assume since you started researching Huntington's at the very beginning. And what have we learned about these mechanisms? I know that you recently published a paper that found links between metabolism changes in certain brain cells with Huntington's. Can you tell us more about these mechanisms?
Sure. Yes. So speaking of that particular paper that we just published, I think it's been interesting because before the gene was cloned, it was thought that Huntington's is a mitochondrial type disease. There's clear changes in metabolism, in the production of energy by mitochondria, which is the powerhouse of the cell. And before the gene was identified, there were attempts to treat the disease with things like coenzyme Q, which is thought to ramp up some of the capacity of the mitochondria to produce energy. And so it's interesting when now we've gone sort of full circle with a couple of our studies recently, where it really is, metabolism is a big piece of this that is dysregulated. So for instance, from that study, we found, using a really amazing technology where you can look at individual cells in the brain and identify the RNAs that are changed. So it's called single nuclei RNA-seq, or RNA sequencing. And you isolate individual nuclei from each cell in the brain, and each one of those is its own little population that you're going to examine the RNA expression in. So you can look at all the neurons in the brain. You can look at the astrocytes or the microglia. So you can get a picture of how each cell is responding to the presence of the mutant Huntington. And so from that, we had found that several of these cells didn't have their correct profile, and some of what looked to be regulating some of the deficits were metabolic changes. And then the Ryan Lim, who was the lead on that, looked at some of the enzyme that might be driving that, and it turned out that there's deficits that cause a disease that's similar to HD, and could be treated by high levels of biotin and thiamine, which are metabolites, which are things that you can get in the diet. But so it kind of came full circle back to this idea of metabolic changes that are elicited by mutant Huntington. And we also found in human patient iPS cells, inducible pluripotent stem cells that are driven to a neuronal population to look like neurons, that even in those cells, when you look at them at a very, very deep level with this very sophisticated type of microscopy, you could actually see changes in the mitochondria that were different from HD patients versus unaffected individuals. So that's kind of one area. I've been working with Joan Steffen for a very, very long time, who's a colleague here and part of the group. And she, one of the early findings that we had in the lab was about the interactions of Huntington with various what are called transcription factors. So factors that drive gene expression and interactions with proteins that become very important for what Huntington normally does. For instance, she had found that this is involved it's involved in protein clearance, so clearing the trash out of the cell by a process called autophagy, that Huntington is intimately involved in that, the normal protein, and that gets affected in HD. We had found that there's tags that are added onto proteins, including Huntington, that drive where they go in a cell, how they function, how they're cleared. And so we've been studying that process for many years. And interestingly, that is now integrated with some of the findings around DNA damage repair and age of onset modifiers. So now things that we've been working on for the last 10, 15 years have emerged as fitting into these same pathways that are involved in modifying when an individual manifests Huntington's disease. So it's been really fun to see how the intersections over the years of the different, areas, mechanisms we've been studying and others in the field have been studying, and how those are starting to come together, how there's findings. A former graduate student here at Ty Nguyen has found that a hallmark feature in ALS and frontal temporal dementia is also found in Huntington's disease and seems to be regulating some of the gene expression changes that happen. So we're building from information from other fields, learning from our own and other people's research in Huntington's disease to understand better, the mechanisms and therapeutic strategies.
That's great. And you mentioned these mechanisms. What insights from learning about these mechanisms can inform the development of effective treatments? And you might have mentioned one potential area, which are the metabolites. But have there been others? What kind of treatments can we look for?
Definitely. So, for instance, there's been quite a bit of effort to try to reduce the level of Huntington, the mutant protein, and that's well underway. We're learning from that we do seem to need some of the normal function there, and so you really want to potentially target the mutant form. We also know from this whole area of modifiers, the DNA damage response, that there are companies, there are labs that are trying to develop drugs around those findings. So understanding that modifiers involved in DNA damage repair are important has now driven a whole area of therapeutic development for Huntington's disease. And we'll see how that works out, but at least when you modulate it or tweak it genetically, it seems to have an effect, say, in a mouse model of Huntington's disease. We're also learning, as I mentioned, from these post-translational modifications or these tags that can get added onto Huntington and other proteins, that might be another therapeutic target. We're developing a variety of approaches to do that, to increase some of the processes that have been discovered, like protein clearance, that are decreased in their efficiency in HD, to potentially see if there's ways to increase their effectiveness. So the more we learn about the mechanisms, the more targeted the development of therapies are. For many years, it was really sort of shots in the dark of a drug that might, it might have some effect on the cell, but we weren't targeting a specific pathway or enzyme or mechanism. And now these efforts are targeting those processes the more we learn. And as we go along, some of these clinical trials are not going to work, but we will learn from them what might be most predictive, say in a cell model or a mouse model, that might inform something that will work, for instance. So it's this constant back and forth between mechanisms, understanding mechanisms, and trying to learn from the clinical aspects of the disease.
So do you think that we're very close to getting some treatments that might show some effectiveness? Or
I always hesitate to answer that question because it's been 30 years since we identified the gene, and we really thought when that was identified we would have a treatment in 5 years, right? And it's been 30 years. The brain is so complex. The ability to get things into the brain, to understand how a treatment might impact, and because we don't have something yet that has changed the course of the disease, we don't know what outcomes to look for in model systems, for instance. So, but we know so much more than we did, and there are approaches that I think, if nothing else, may modify disease as we go along. I don't know about cure yet, but I think it's in the near future. It's just drug development takes many, many years. We're trying to develop a stem cell based treatment for Huntington's disease, a transplantation based mechanism, but it takes years in development to identify a treatment, and then all the various stages of clinical trials that you need to go through to make sure things are safe before you even look at if they're effective, for instance. So I have quite a bit of hope that it's in the near future, but wouldn't give a timeline.
Great. And we look forward to many coming discoveries, and I'm sure that your lab is, and many other labs are working on it.
Yes, and the thing about HD that I love personally is it's a very collaborative field. We all know this is hard. It's a very difficult disease. We're very close to the families and the patients and all of us, and we want something. So people tend to work very closely together, share information, and okay, you have this tool, let's do this. We have this tool, let's put this together and work together on it. And that helps a lot.
Mm-hmm. And we have another audience question here. What is the lifespan of a typical HD patient?
It can vary. So let's say somebody has 42 repeats and they start showing disease at age 40. So they start showing some chorea. Typically, the progression of this disease as they get kind of worse and worse is about 15 to 20 years after onset of symptoms. So it takes a very long time, unlike ALS, which typically will, once onset has happened, it's maybe 2 to 3 years of till someone passes away. For Huntington's disease, they typically will pass away from aspiration pneumonia or a fall, hit their head, cardiac event. So it's from the consequences of Huntington's disease, but it's a very long, difficult process.
Mm-hmm. And what excites you most about your research moving forward, and what do you hope to achieve in the coming years?
Well, I'm very much hoping to find something that will change the course of disease. We have several different ways we're trying to do that. One of these is this, what I was mentioning, these tags, these tags that go onto proteins and are also involved in DNA damage repair. And the stem cell based treatment, but which we're very hopeful for. But I think what really excites me is the way our understanding of Huntington's disease, ours being global ours, has been evolving, and how things that we didn't even realize sort of met each other in terms of the research are now beginning to become integrated. And I see that in my own lab, where multiple projects that seemed very distinct initially are informing each other, and I think will help lead to a treatment. So for me, I mean, that's what I want to see more than anything, and what I hope to see in my lifetime, my career lifetime, I guess I should say.
Great. And anyone who is watching this podcast and is a young researcher and wants to know and learn how they can get and pursue a career in neuroscience, possibly doing research into neurodegenerative diseases, what would be your advice for them?
I think it would be, some of the usual things. You would take courses in neuroscience and maybe some in neurodegenerative diseases. I would recommend as an undergrad, you do work in a lab, do an undergraduate research project. And one thing that's nice, I think, that maybe people don't think about sometimes is, you can do anything. You don't. And if I started out as a yeast molecular geneticist. So I was working in the yeast field on metabolism as a graduate student, and became very interested in neuroscience, and then did a postdoctoral fellowship in that area, in neurodegenerative disease. So you wouldn't have necessarily anticipated that transition, but you can do things, and you can apply the tools that you've learned in any field. We applied tools that we learned in the yeast field to our work on Huntington's disease, which might sound a little surprising, but it's really helped. And so I think that surrounding yourself with mentors, reaching out, but not being afraid to do collaborations, reaching out to other groups to work together, and also being willing to do new things, embrace new technologies. It's sometimes scary to jump off that cliff of taking on a new technology or a new approach, but it's very rewarding in the end.
Great. Well, thank you so much for your great advice. I'm sure others will watch this and make full use of that. And then finally, I read somewhere that your favorite quote is by Eleanor Roosevelt. And I'm going to read this quote. It says, "We gain strength and courage and confidence by each experience in which we really stop to look fear in the face." Dr. Thompson, can you share a specific experience in your research or in your life where you had to look fear in the face, and how that helped you to grow and develop as a scientist?
Sure. There's been many times in my personal life that has applied. One time that sort of integrates the two, the personal and the scientific aspect, would be when I was a postdoctoral fellow. My advisor passed away when I was a senior postdoc. And at the time I had 2 young children, and I was kind of working semi-part-time at the time. I wasn't anticipating going down this route of becoming, a professor and having a lab and doing this kind of research. I really wasn't sure what I was going to do. And at that point, I had to make a decision of, I either need to go for it and sort of take over leadership of this project of the Huntington's disease work from the lab, or not. And so that was one of those times, definitely being scared to death and looking fear in the face, not knowing if I could really do it. It was a big decision. So that was a pivotal time when that quote definitely applied. And there's been many other times, even more minor things, like I was talking about embracing a new technology. There was this time when RNA sequencing was emerging, and these technologies, and it was pretty intimidating to start jumping into that area. And I took a sabbatical and started to learn more about it. And so that was another time just to say, okay, let's do this. Let's learn about it. Let's bring it into the lab.
Well, thank you so much, Dr. Thompson, for sharing that experience with us. And thank you for joining us today. And how can we learn more about your research, Dr. Thompson?
Publications on PubMed, or we're not the greatest about social media, I will confess. But there are occasionally things, press releases that come out of, and information from UCI on the research. Our website, to some degree, needs to be updated, but so those kinds of areas, and always happy to. We have, I guess, one way, though, we have many events. We have a group here that's called HD Care on campus that's part of UCI Mind, actually. And it's a Huntington's disease support group that's part of UCI. It's a UCI support group. And we often have educational events, and we distribute information about those. And also, we are at Huntington's Disease Society of America Center of Excellence here at UCI, with both the clinical arm and the research arm, and again, have frequent educational events. We sometimes have tours of the lab, so we try to reach out to the community and provide information about the research going on here and elsewhere.
Great. Well, thank you. Thank you so much for, again, joining us today on Ask the Doc. And thank you all who have attended this live session today. You can access this and past Ask the Doc episodes and podcasts to learn more. We can provide you with our website, which is mind.uci.edu/Mindcast. And next month, we'll be chatting with Dr. Liz Tracedill about spatial navigation. And in the meantime, to learn more about UCI Mind, again, our website is mind.uci.edu. And please take care, and we'll see you next month for Ask the Doc on UCI Mind.
Thank you.